it's basically this idea that you want to be able to target these viruses and essentially prevent them from entering the cell, okay? But in order to build to the drug discovery, and also just being the first in this COVID-19 lecture, I felt it was necessary to just start with an overall background of COVID-19. Let's see if I can share the screen yet. No, I cannot. So yeah, sorry about all this, but it's the first one. We're working out the kinks. So just kind of with my presentation, so the whole idea behind this entire series is this idea, here we go. Thank you. Okay, now we can see the screen. All right. So the whole entire idea behind this series is to build off of Alexander Campbell's philosophies. And that's the idea to free the human mind from vulgar prejudices, ignorance, and error, right? And so for this talk, for this series, we're essentially providing that knowledge. So there's a lot of knowledge out there. There's a lot of, incorrect knowledge that is spread. So we see this with Facebook, we see this with Twitter, we see with all these social medias and the different news outlets. So it's hard to know what is real and what is not. And so the entire point of this series is to help free your mind and learn what actually is true about COVID-19. And so that also builds upon, so Larry Grimes also said that essentially we want to take this knowledge and we want to take this idea from Alexander Campbell built upon the Bethany seal, and we want to take that truth that we learn and we want to put that truth into action, not just into theory. Okay. So for my talk, I'm going to start with what's a name. So what does COVID-19 actually mean? What is COVID-19? Where does it fall in terms of diseases and illnesses? Then kind of just a brief introduction on what is a pandemic versus an epidemic. And then we'll lead into, so SARS-CoV-2 is the actual virus that causes COVID-19. COVID-19 is the disease. We'll talk a little bit about SARS-CoV-2 and other coronaviruses. We'll then go into the viral transmission, so how the virus actually enters the cell, which again is essential for the drug discovery process. And then we'll talk a little bit about our testing procedures and the drug discovery process. There is a lot in this presentation. I hope we can get through it all, and I will do my best. All right, so what is COVID-19? So what does even COVID-19 stand for? So we have two different parts of COVID-19, and we have the actual virus itself, and that virus that infects our cells is the severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2 for short. Okay, you will remember, and we'll talk about in a minute, that SARS was a epidemic, a virus that was an epidemic in 2002. And so this is actually the second strand, it's a sister strand of that SARS virus. And so this is the second one. The disease itself, so once the virus infects your cells, the disease itself is what we call COVID-19. And COVID-19 stands for, so the CO stands for corona, the VI stands for virus, the D stands for disease, and the 19 stands for 2019. So while this epidemic and pandemic of COVID-19 has actually taken place in the majority of 2020, the actual virus itself, the first disease, was in late December of 2019. So that's why it's COVID-19 and not COVID-20. And so it's really important to realize that this is the actual name of, so the SARS-CoV-2 is the name of the virus, and COVID-19 is the name of the disease. So according to the World Health Organization, the CDC, and the National Institute of Health, so moving forward, we are no longer going to name epidemics and pandemics by their geographical location. Okay, so by naming them by their geographical location, one thing we have seen with COVID-19 is it presented this inherent racism towards individuals that are from that origin area. Okay, but it's also from the fact that the origin area isn't always accurate. The Spanish flu, for example, was actually believed to originate in the United States. It was named the Spanish flu because Spain was the location that had the most number of deaths associated with the flu virus. Okay, so honestly, if we were at this point in time with the statistics, if we were to name COVID-19 that way, it would be the American flu, not the Chinese virus. So we no longer use those names because it just presents that inherent racism and bias towards those countries. And exactly with what Alexander Campbell said, that's what we're trying to avoid. So it's really important that you use the SARS-CoV-2 nomenclature when you're talking about the virus, and COVID-19 when you're talking about the disease. All right, so what is COVID-19? Okay, so COVID-19 actually is a coronavirus, as we all know, but this is a family of virus. So we talk about COVID-19 as a novel coronavirus. Doesn't mean that it was the first one. It just means that it's a new coronavirus, it's a new strain, a new virus that we haven't seen before. Okay, there's actually a large family of viruses, and they're all characterized by this crown shape. So corona is Latin for crown. And so you actually have these protein spikes on the surface of the coronavirus. So if you kind of draw around those protein spikes, it literally looks like the sun. So the corona of the sun, or it looks like a crown. Okay, so that's where the family of these viruses came from. The first coronavirus was discovered in 1931. The first human coronavirus was discovered in 1962. So the coronaviruses actually infect a wide variety of animals and species. You actually find the most number of coronaviruses present in bats. So there are actually more than 20 different coronavirus strands prior to SARS-CoV-2. One thing that these coronaviruses do have in common, so as they transfer into humans, they are what we call zoonotic diseases. So zoonotic diseases are any disease that basically is able to mutate and transfer from other species into humans. This figure over here shows you some other zoonotic diseases that we are very familiar with. Lyme's disease, for example, Ebola, SARS. SARS is another coronavirus. E. coli, chickenpox or cowpox, rabies, tuberculosis. All of these are diseases that originated in other animals, other species, but through mutations, they were able to jump into humans. And so now we are also get sick from these viruses and bacteria. Okay, SARS-CoV-2 is actually the 7th zoonotic coronavirus to date. So there are 6 other coronaviruses that we have actually seen in the human body. Okay, so those 7 that we know of, there are ones that are low pathogenic coronaviruses. And so these are viruses that can cause disease, but the disease isn't necessarily detrimental. These types of coronaviruses actually cause the common cold. There's 4 different strands, and you can see here when they were discovered, so in 1962, 2004, 2004, and 1965. Okay. The majority of these coronaviruses, you can see, originated in bats. There are some that originated from rodents, but essentially what happens is over time, those viruses mutate, and then they're able to jump over to these other species. So in the case of our highly pathogenic coronaviruses, so these are diseases that have actually caused pretty significant illness and ultimately result in death. With the common cold, with the low pathogenic coronaviruses, you actually don't see much death. There would be pretty significant underlying conditions for someone to die from a common cold. Okay, but with the highly pathogenic COVID coronaviruses, there's the SARS-CoV-1, which was the SARS outbreak between 2002 and 2004. It passed from bats to civet cats to humans. There's the MERS-CoV from 2012 that went from bats to the dromedary camels and then into humans. And now we're experiencing SARS-CoV-2, which was transmitted from bats. It was believed to be transmitted from bats to pangolins. The question mark is because it's not absolutely certain that that's the method, but that's the method we believe it occurred by. And so then from pangolins into humans. Okay, so with the coronaviruses, so for a second, I'm going to compare the COVID-19 to the SARS and the MERS outbreaks that we saw. And to really understand the differences, we need to understand the difference between what is an endemic, what is an epidemic, and what is a pandemic. Okay, so those of you that are Bethanians or and alumni, you are very familiar with an endemic. So any time in the winter that you experienced what we call the Bethany plague, right? So anytime there was just a outbreak of disease in Bethany, in the Bethany bubble, that's what we call an endemic. So this is an infection in a population. That infection is constantly maintained, and it has a baseline level, and it's localized to a specific geographical area. So you may have endemics in multiple areas around the globe, but they are localized to those specific areas. Then there's the epidemic. So an epidemic is similar to an endemic, but with an epidemic, it's a rapid spread of the disease. So you have this very quick upshot of this disease, and everybody, essentially everybody within a population gets that disease within a short period of time. So here, the epidemic, you have more people, you're still localized within one region, or it could be a couple of regions around the globe, but you're still localized within one region, and it's really quick upshot of this disease, and everyone gets sick in a very short period of time. So the coronavirus, so COVID-19 started as an epidemic in China, and then over time, as it spread across the globe, in March of 2020, it became a pandemic. And so a pandemic is an epidemic when it spreads to many other regions across the globe. And it's quite interesting because pandemics we have some pandemics throughout history, but with the evolution of technology and the invention of the airplane, we're actually more prone to the epidemics evolving into pandemics because of just the rapid travel that we have getting across the globe, right? So you can prior to the 1900s, it would take you a month at least to get from one side of the globe to the other. Now you can be across the globe in a single day. So if you are carrying the virus and you're sick, but you don't know you're sick, you will be at the other location across the globe while you're still contagious. And that's something that's kind of a newer evolutionary phenomenon that has developed with the advent of air travel. So kind of talking about the three different coronaviruses that we most commonly know, these highly pathogenic coronaviruses. The first one was the severe acute respiratory syndrome, or SARS. This was an epidemic from 2002 to 2004. It reached a total of 29 countries. So since it didn't spread across the globe, it remained an epidemic. With SARS, there was a reported 8,437 cases with a total of 813 deaths. That works out to be a 10% death rate, excuse me. The incubation period, so this is the really key thing to keep in mind. So when comparing SARS to MERS to COVID-19 or to the SARS-CoV-2, what really comes into play is the death rate and the incubation period. Okay, so with SARS, there was a 10% death rate, and that incubation period was 2 to 7 days. And the most important key thing to note is that people were only contagious with SARS when they had symptoms, right? So with SARS, if you started to express symptoms, you weren't going to take that trip across the globe, right? You were going to stay home, you were going to get better. So you were only contagious when you had those symptoms. Okay, so that's a big difference between SARS-CoV and SARS-CoV-2 that we'll talk about in a second. The really important thing about SARS was that it was the first severe and readily transmittable disease in the 21st century. And it was SARS that was the first thing to show, so here's a cover on the Newsweek, that it showed this capacity to spread along these routes of international travel very quickly. So this was that first red flag to say, hey, with this international air travel that we have, people are far more at risk for the development of a pandemic than an epidemic. And so SARS was the first indication that these global pandemics were something to be worried about. So MERS, which is the Middle East Respiratory Syndrome, so it's MERS-CoV, also coronavirus. So you can see here, so this is a picture of MERS right here. So you can see that coronavirus shape for that virus. MERS was also an epidemic. It only reached 27 countries. And it's actually interesting with MERS, it was actually the death rate and the fast incubation period that prevented MERS from spreading globally. Okay, so there were but there were 2,519 cases. And of those cases, there were 866 deaths, which gave you a 35% death rate. So a very deadly disease, and a very fast disease. So the incubation period for MERS was within about 4 to 5 days, you had symptoms, and with those symptoms, you were immediately hospitalized within 4 days. And those that died from the virus were dead within 12 days. So because of this very quick incubation period and the high death rate, the virus, so MERS, was not able to spread very far just because people got sick so fast and they died so fast they did not have time to spread the virus. And so with COVID-19, so the virus again is SARS-CoV-2, so severe acute respiratory syndrome coronavirus 2. With SARS-CoV-2, it's the first of these coronaviruses to reach pandemic status, and it did so in March of 2020 of this year. It has reached, so comparably to the 29 and 27 countries that we saw for SARS and MERS, coronavirus has reached 188 countries and regions. It has spread to the majority of the globe. And as of yesterday, there was a report of 27,249,308 cases with 890,971 deaths. This gives you a death rate of 3.3%. So the death rate is lower with COVID-19 than it was with SARS or MERS, but the difference is the incubation period. So with the incubation period, so they're still working on investigating the incubation period, and they've narrowed it down so it can be anywhere from 2 to 20 days. So we typically talk about the 2 to 14 days. So someone needs to quarantine for 14 days if they believe they've been infected, right? But we're still kind of narrowing down that incubation period. But the biggest difference with COVID-19 is that asymptomatic individuals are contagious, right? So even before you start to feel sick, you are carrying this virus and you are spreading this virus. So essentially what happened is at the start of the virus, people felt fine, and they still do, and they're going out, they're traveling, but when they were traveling, they didn't know they had the virus until they landed in the other location that they were going to, right? So it allowed for that global spread, and the virus just kind of hides within individuals. And that's why we've seen such a wide spread and such a larger increase in cases from the other two coronavirus strands. Okay, so this map just kind of gives you an idea. It really just puts it into perspective. With the SARS-CoV, so the SARS in 2002, you could see, so very small. So the size of the dot is indicative of how many cases there were and where the cases are located. So you can see how small the SARS virus from 2002 is compared to the SARS-CoV-2, where you have all these dots in every country, very large dots completely spread across the globe. So it's very, just seeing this map to me is just amazing to see how far the virus has actually spread. Okay, so that leads us into the idea of how does SARS-CoV-2, how does it actually infect our cells, right? And the reason why we need to know this is because in order to understand and in order to develop drug therapies for these viruses, so these antiviral medications, we have to know how the virus actually works. And so this is the entire idea behind molecular pharmacology. So we're developing drugs based on the pathways and how these molecules work at a microscopic level within the cells. Okay. So with the SARS-CoV-2, much like SARS-CoV, it actually binds to the ACE2 receptor, which is an angiotensin converting enzyme 2. So the viruses, so if you imagine the viruses just kind of floating, so they're spread by aerosol, they're flying through the air, okay, they need something to bind to, they need something to lock onto in order to be able to enter the cell. Otherwise, they're just going to keep flying around. And so it's that ACE2 receptor that's on the surface of the cell that basically serves as a binding site for the virus to bind to. So those spikes that give the coronavirus its actual name and shape come down, they bind to this ACE2 receptor, which we have high expression levels of this ACE2 receptor in our nasal and respiratory passages. And that's what causes this SARS-CoV-2 to be a respiratory disease. There's actually some newer research indicating other activation pathways, but for right now, this is what's known. So the ACE2 receptor is what binds the coronavirus, what binds the SARS-CoV-2. Once it's bound, this other protein, this TMPRSS2, or type 2 transmembrane serine protease, actually cleaves the ACE2 receptor, and then it's that cleavage of the ACE2 receptor that allows the virus to enter the cell. So you have this ACE2 receptor, you have this TMPRSS2, which these are both necessary on the surface of the cell in order for the virus to actually enter the cell. It's actually quite interesting that males express ACE2 significantly higher than females. And so it's actually, there's a correlation that males are more susceptible to being infected by SARS-CoV-2 than females, just because of the expression levels of these ACE2 receptors. Okay, with the ACE2 receptors, so you also can see over here that children express less ACE2 and express less TMPRSS2 than adults. So this is why adults are more susceptible to developing COVID-19 than children. You can also see that ACE2 and the TMPRSS2 expression is increased with smoking and COPD. It's increased in, as I mentioned over here, in adults over children. With asthma, there's an increased expression. With hypertension, there's an increased expression. And so there are actually other factors that play into ACE2 and TMPRSS2 expression levels that can make some individuals more susceptible to developing COVID-19 than others. So once inside the cell, so we also need to know what the virus does once it's inside the cell. And so once it binds to the ACE2 and the TMPRSS2 and it enters the cell, okay, once inside the cell, the virus actually releases its RNA. So this is the viral RNA. That viral RNA is then translated into expressing viral proteins. It is also replicated to make more RNA. And then the combination of those viral proteins and the replication complex come together to make more virus. And then that virus is then released to the cell and then goes and repeats this process to neighboring cells. So it's an exponential process of how the virus infects our body and our cells. Okay. So you can look here. So there are a couple of different ways. So when we talk about drug discovery, ways to prevent, so these antiviral medications that we talk about, they prevent this entry of the virus into the cell. They prevent the expression of the mRNA. They prevent the production of these viral proteins. And so they basically use these complexes and you target these areas to help prevent the virus from even entering the cells. Once the virus enters the cell, and once the viral proteins and the RNA are replicated, then that virus is going to grow and express. But these antiviral medications, how they work is they prevent this process from happening. Okay, I'm actually, for time purposes, so this part of the lecture I was actually going to talk about, so the methods for testing COVID-19. I think I'm going to skip ahead to our drug discovery methods. Real quick, I will go through this very quickly. So there are different types of testing. There's the diagnostic test, which we have molecular testing, which actually tests by PCR. And then there's also antigen testing. So when these viruses essentially take over our cells, they start expressing these antigens or these proteins on the surface of the cell. It's basically the virus's way of sticking the flag in the cell and claiming it as theirs. And so our body actually responds to the presence of these antigens, and that's how we make antibodies, and that's how our immune system responds to the presence of a virus. Okay, so we have these different methods for testing for COVID-19. There's the PCR test, which actually literally looks for the presence of the virus. So it looks for the viral DNA or the viral RNA and sees if it's present in our cells. There's the antigen test, which actually looks for those proteins that the virus expresses on the surface of our cells. And those are the two diagnostic testing. Once you have the virus, there's the serology test or the antibody test. So when you're first infected with the virus and you're contagious, you're going to have antigens, but your body has not yet started producing antibodies against those antigens. So that's why we can't use antibodies in our diagnostic testing. But if you're looking for people that have been infected with the virus and have recovered for the virus, now we can use this antibody test to see how far the virus has actually reached. Okay, so, well, with the molecular testing, so many of us, so those that were on campus the 14th and 15th and had the COVID-19 testing done, you're very familiar with this method, which I really call the picking your brain method. So they actually reached the Q-tip, very long Q-tip, all the way back into the nasopharyngeal region or in the oral pharyngeal region in your nasal passage, so in your throat and nasal passage. And so they're literally going to the very back of your sinus cavity, and they're testing and they're scraping and getting some cells to test for the presence of COVID-19. Okay, with the overall molecular testing process, it's important to understand that this testing process takes a long time. Okay, so that's the big downfall of this test. So it takes anywhere from 5 minutes, depending on how long you have to wait in line, to a few hours to take the sample. Once this, and there's other methods to take the sample. There's also, you don't have to go quite as deep in the nasal cavity. So there's the nasal mid-turbinate area. So it's still a deep nasal swab, but it's not as deep as the nasopharyngeal swab. You can also have the anterior nares specimen, where they just do circular motion right inside of the nostrils. In more severe cases, they can do a nasopharyngeal wash, where they essentially spray saline up through your nose into your sinus cavity, and then when that saline comes back out, it actually will contain cells that in theory would contain the virus. In the most severe cases, they can do a lower respiratory specimen, where they literally go in and do a lung biopsy. Okay, so once they have the samples, it takes about 24 to 48 hours for the samples to be transported from your testing site to the testing facility. Most tests are analyzed at the CDC. There are some companies that have private testing sites that do testing on their own, but for the most part, the CDC are the ones analyzing these samples. And that process of getting to the CDC from your testing site can take anywhere from 24 to 48 hours. Okay. Once at the testing site, they need to isolate the viral RNA, and there are kits that do this for you. You actually have what's called column chromatography. So this is a silica column that essentially binds everything except for the viral RNA, and it allows you to isolate the viral RNA from the rest of, so for example, your mucus, your cells. And so it ensures that the only thing that they're collecting, the only thing that they're testing is that viral RNA. Once they have the viral RNA, they do what's called a reverse transcribe. So they convert the viral RNA back into DNA through an enzyme called reverse transcriptase. And so that actually gives them the DNA code, which it's called the cDNA, or the coding DNA. And this is what allows them to perform the PCR, or the polymerase chain reaction. And so what polymerase chain reaction, what PCR is, it actually allows you, so it's an amplifying process. So you take you can have as little as one strand of DNA, and over time through DNA replication, it actually duplicates the amount of DNA you have with each cycle. So it's an exponential increase in the amount of DNA that you're actually expressing. So, and it specifically expresses the viral DNA. So we've targeted the viral DNA. We actually put what we call a fluorophore. We attach a fluorophore to this DNA, which basically says that if that DNA is present, the fluorophore is going to light up and basically express this fluorescence signal. If the viral DNA is not there, there will be no expression. And so you can actually, so if you repeat this cycle 40 times, you actually end up with 1.1 trillion copies of that single DNA. This is the same procedure they use at crime scenes. So if they're analyzing DNA at crime scenes, they use this polymerase chain reaction, this PCR, to amplify the DNA that you have and express. But in this case, we're using it to see if we have the presence or absence of this virus. And so someone that is positive for the SARS-CoV-2 is going to have a high expression level. So it's that fluorescence is basically going to light up and you'll be able to read that expression. If you're negative, so if you don't have the DNA of interest, you're not going to have any amplification. You're not going to have any fluorophores lighting up, and therefore there's not going to be any expression of this DNA. Okay, and so that's how you analyze between positive and negative. Overall, with the viral isolation, the reverse transcriptase, and the polymerase chain reaction, that takes about 5 to 6 hours for that entire process to occur. The part that people don't take in consideration is once you have that data, you have technicians that then have to take the individual tests and they then have to do all the computational analysis. So they have to correlate, okay, this test was positive, correlate it with this ID number that came in for that sample. And you have to basically reestablish that connection of which sample goes with which person. And that process can take anywhere from 24 to 48 hours. So that's why all of this taken together, that's why this molecular test, so the time it takes to travel to the lab, the time it takes for the lab to actually perform the assay, for the time it takes to actually type in the data and report that assay back, it takes about 5 days in order for all this to occur. And so that's why it takes so long to get your test results back. Okay, and just so I have a friend, her name's Heather Greiser. We used to work at the Drug Discovery Institute in Pittsburgh together. She actually works in the main CDC now, and I asked her to give us some idea of, you know, what actually is happening in the CDC. And so on any one day, so this is a picture of her desk over here, and these are results that she has faxed to different testing sites, to different so hospitals, different individuals, with their results, right? So this is just a week's worth of paper. Also, they're able to run anywhere from 1,200 to 1,500 samples a day. When they run those samples, they're running new samples that they've received that day. They're rerunning samples that failed for various reasons. They're also, so people that test positive, they then go back and repeat those single samples as needed. But then the data entry, so if you see here, so this was actually one day where her team completed over 1,500 assays in one day, and they were waiting for the data entry to occur. And the data entry, you still had to input this 1,300 results within a day. And so there's a lot, so essentially these individuals in the CDC are working constantly to spit out this data, but it just goes with this philosophy that science takes time. And it doesn't do any good to rush science. If you rush science, you increase the chance of messing up. So, so it takes a long time, not to mention the fact that the whole time they're also fielding hours worth of phone calls daily from epidemiologists across the country. They're coordinating outbreak sites. They are checking, so people that are calling to check on their results. So they're constantly taking these phone calls on top of their daily testing, and they've been doing this every day since March. So these are the true heroes. So the health professionals and the people working at the CDC, these are the true heroes behind COVID-19. So if you ever see somebody who works for the CDC, please thank them for me. So the last part of my talk goes into talking about, how else, oh, wait, sorry, I'm sorry, I apologize. So this is the antigen test, I apologize. So the antigen test, it works similar, but it uses what we call the ELISA assay, or the enzyme-linked immunosorbent assay. The ELISA assay actually looks for these antigens that are on the surface, so these proteins that are expressed specifically by the coronavirus, by the COVID-19, and you essentially have a plate, and on that plate you have what we call a capture antibody, an antibody that specifically targets the antigens on the surface of the virus. When that antibody binds the antigen, there's then a second antibody that then binds to the other side of the antigen, and then that second antibody actually has a fluorophore attached to it, which then expresses a signal. So if you have the presence of this coronavirus, if you have the COVID-19, if you have the SARS-CoV-2 antigen, it's going to bind to the capture antibody, and then the second antibody is then going to bind to the antigen, and then that's going to light up as well. If you don't have the antibody, then nothing binds, and there's no fluorescent signal. Okay. The advantage of the ELISA assay is the assay is completed in 30 minutes. The disadvantage of the ELISA assay is you do need a baseline expression level of the antigen. Whereas with the PCR method, all you needed was one strand of DNA. With the ELISA assay, you actually need at least a certain concentration of the antigen in order for the assay to work. And then the last test is the serology test. So this is actually looking for the presence or absence of antibodies. So essentially after you've had the virus, you've developed these antibodies in your immune system. And so you're essentially looking for the presence or absence of the antibody. Okay, these tests work a lot like your, say your blood sugar. So with diabetes and testing your blood sugar levels. So it takes a single prick of the finger, and then you put that blood on the testing system, and then it detects the presence of it tests for the presence of two different antibodies that would be expressed if you had the COVID-19, right? So you have a control. So the control here basically tells you that the assay is working, and then you can either have just the IgM antibody, you could have just the IgG antibody, or you could have both. And so these are antibodies that are specific to COVID-19. And so this is, again, an antibody test, or the serology test. Okay, so the last thing to talk about, as we're kind of running out of time, but we have the FDA drug discovery process. The big problem with the drug discovery process is, so we're dealing with a pandemic, and we need medications fast, right? So in order to develop any new medication for any disease, it takes, so the basic research behind it takes 2 to 3 years. Non-clinical studies takes 3 to 5 years. So you're taking anywhere up to 8 years before you're even getting into clinical trials. So right now, the process that we're taking is we're taking medications, we're taking other antiviral medications, and that are used for malaria, that are used for the flu, that are used for, that were previously used for SARS. And we're using these antiviral medications, and we're essentially taking them and we're throwing them into these COVID-19 clinical studies, okay? There's 3 phases of our clinical studies. There's phase 1. In phase 1, they're basically just testing the safety of the medication, making sure that it's not going to harm any of the patients. In the phase 2, they're upping the size of participants, going from like 20 to 80 participants in phase 1 to now 100 to 300 participants in phase 2. And now they're testing how effective is the drug, how safe is the drug, and at what concentration can we actually give this drug. So once they approve phase 2, then they move up to phase 3. Phase 3 is the same as phase 2. You're just now testing on more participants. Okay. Once a drug completes phase 3, you then have a 1-year trial period, like you're distributing that drug, but you're making sure you're keeping a close eye on the drug, making sure that there are not any really bad, harmful adverse effects of the drug. And that takes another year. So if you look at this time frame, we're trying to squeeze this drug discovery process in as short a time as possible with this COVID-19 disease. And so again, this kind of shows you that with the SARS-CoV, it goes back to that image before. to where we want to try to find ways, so where the drugs that we're using target, prevent the binding to the ACE2 receptor, which prevents the internalization of the drug, of the virus. If the drug is able to enter still, then you can also use other drugs to prevent this membrane fusion, which is where the virus releases the RNA. If the virus cannot release the RNA, the virus cannot replicate and it cannot continue to spread. So we're trying to use previous medications in this signaling pathway to see if we can apply other antiviral drugs to the SARS-CoV-2 virus. And so some of those medications that we've used before, so we've heard about the chloroquine, the hydroxychloroquine, but with the chloroquine, the hydroxychloroquine, also with the lopinavir and the ritonavir, both of those, all of those medications have already been disproved by the CDC and the FDA and are not effective against COVID-19. There are other medications that you can see that work on this antiviral pathway. And there's one, so that there's the Camostat Milosate, which is currently in clinical trials. So that is a molecule that blocks the TMPRSS2. If you remember, the TMPRSS2 is necessary for the drug to actually, or for the virus to actually enter the cell. Okay. There's also this remdesivir, which is actually has shown promising effects. It has been shown in phase 1 trials to slow or to speed up the recovery process. So patients actually recovered faster from the virus. So that's been promising in phase 1 trials. And then we also have this idea of the convalescent plasma, which convalescent plasma is essentially, they're taking plasma, they're taking antibodies from people that have recovered from COVID-19, and they're injecting those antibodies into people that are currently sick. And it's kind of used as a way to essentially jumpstart the immune system. These methods, so the convalescent plasma is not approved by the FDA, but it is regulated as an investigational product. So they're allowing people to try using this convalescent plasma, but they're keeping a really close eye on it. Okay, the other way to target COVID-19 that we've heard about is the vaccine development. So the previous slides were talking about drugs to actually target the virus once infected to prevent future spread of the virus. You can also use the immune system. And so with these vaccines, essentially what the vaccine does is it puts up a wanted poster in your body, and it alerts your body that, hey, if you ever see this SARS-CoV-2 virus, you need to kill it immediately. It's a bad guy. Okay. In April 2020, so in April, there were 114 vaccines that were in trial. There were 91 that were exploratory, 18 that were preclinical, and 5 that were in phase 1. At this point, as of September 3rd, so there is a website, this COVID-19 vaccine tracker. You can go to that website and check on the updates of vaccines at any point in time. We currently still have 45 vaccines in clinical development. We have 1 that is exploratory, 16 that are preclinical, 9 in phase 1, 8 that are in middle of phase 1 and phase 2, 4 in phase 2, 2 between phase 2 and phase 3. And we actually have 5 vaccines that are currently in phase 3 trials. Okay, so they're still not approved by the FDA, but they're very close to being approved by the FDA. And so we have 5 of those that are very promising vaccines against COVID-19 at this point in time. Okay, so in conclusion, so the overall idea is that you can't rush science, right? So we're in a pandemic and we're trying to find these solutions to this COVID-19 pandemic, but you can't rush the science. But according to Anthony Fauci, so we're at a point to where, so with the antiviral medications we already know, with the vaccines that are already in development, so you can't rush science, but once you can look at kind of the past of the science. So we can play off of the SARS-CoV virus from 2002 and kind of use that as a starting ground for this SARS-CoV-2. And so once that science points you in that direction, so once we knew that this was a SARS coronavirus, we could actually then use the knowledge we already had to start rushing towards developing medications and cures for this COVID-19. Other things to keep in mind, COVID-19 is not our first zoonotic coronavirus, and it will not be our last. right? So there is potential that over time, we do have another outbreak of this sort. So we always have that threat of these zoonotic diseases crossing from animals to humans. And so it's always something that should be in the back of our mind. When you do the molecular testing, be patient when you're expecting your results. They do take 3 to 5 days. That's being pretty optimistic, but with this PCR test, your results are there. It's the golden standard for testing for COVID-19. Also, so the FDA drug trials, they take time and for good reason. So you don't want to rush a drug trial process, right? We currently have 5 promising vaccines in phase 3 trials, but we don't want to rush them to the market because you want to make sure, you want to test for side effects. You want to test for drug-drug interactions. So you want to make sure that these viruses are safe, or these vaccines are safe before you distribute them to the masses, okay? And so that is the conclusion of my talk. Hopefully you learned something. Sorry I went over time a little bit, but just make sure, remember to return next week, same time, same place. And as always, wear your masks, wash your hands, and save lives, and protect the herd. I want to thank Dr. Rodenberg, Dr. Lane, Malachi, and Roan for putting this together. I also want to thank Karen Cormuth, our assistant biology professor at Bethany, Jen Franco, who's a past biology professor from Bethany, and then also Heather Greiser, who works at the CDC, for their guidance and help with this talk. Thank you.
Do we have time to take just a couple questions before the hour, Carolyn? Are you game?
Sure, yeah, absolutely.
Okay, we got a couple in the chat room. Do you want to share a couple of those and put them out there?
Sure. Yeah, so Lee and Sally from class of 72 and 71, Dr. Kitchens, have a couple questions. Their first question was, would the plagues of the Middle Ages have been epidemics or pandemics?
That's a good question. So I know our colleague Ian, he's actually going to talk about the history of plagues throughout time. So some of them, so the bubonic plague was believed as a pandemic. Because it actually traveled from China to Europe. So it was termed to be a pandemic. Was that sorry? Yes, so that would have been the plague. So the bubonic plague would have been the plagues in the Middle Ages. And yes, they were pandemics because at that time, so the Western Hemisphere was actually kind of the majority of history that we knew about. And so that was actually considered a pandemic because it went across and encompassed the entire Western Hemisphere.
Okay. They also were asking, how long can a COVID-19 virus survive without a cell host?
So that's the question, right? And that's always the debate. So the CDC is constantly updating their knowledge, right? So at this point, so initially it was thought that the virus could survive on cardboard boxes for, up to 72 hours. But now it's this idea that it's more of the respiratory, so the aerosol distribution of the virus, that is the contagious one. It's not so much, so the surface contact, you still need to worry a little bit, like don't lick a desk and then lick your finger, right? But by limiting touching your face with your hands, by limiting that surface to face contact, you're actually decreasing the amount of time, or you're preventing that surface contact. And so really the big concern is that aerosol spray. So I know that really doesn't answer your question, but it's kind of constantly changing. I'm not sure of the final numbers that they have come to with that, but I know most viruses can survive at least 24 hours on a surface. But as long as you're not touching the surface and then directly touching your face, you should be fine.
Okay, great. Are there any other questions? I think those are a couple of the only ones. Several people really enjoyed this, Dr. Kitchens, and said thank you and everything there in the chat, as you can probably see. Ian's also on this call and did give the correct thumbs up on the first question. And reminder, like you said, Ian will be also giving one of these lectures on November 17th, I believe, covering more of the history side.
Yeah, and so also Dr. Cormuth responded to the question about the surface. So saying, so it depends on the environment. So temperature, humidity, surface material. So there are a lot of factors that go into this. How long can a virus survive on a surface? But the basic idea is, as long as you are washing your hands, you should be fine. Washing your hands and not directly touching your face, you should be fine.
Right, absolutely. All right. Any other questions? Everyone can feel free to put them in the chat.
And then also, you can also, so if you wish to ask me more questions, especially about the drug discovery process, I know I really wanted to focus on giving an introduction to coronavirus for every other talk. I could give another talk on just drug discovery and viral vaccine research. But if you have any questions, you can email me. My email is ckitchens@bethanywv.edu.
Well, thank you very much, Carolyn. Thank you for everybody who's joined us. We certainly hope to see you back next week when